Photosynthesis. All right. So photosynthesis is the biochemical process by which plants will capture energy from sunlight and convert it into carbohydrates. This roughly translates into something using light to make sugar. Usually plants, which we'll be talking about in this section a lot, are going to take up water through their roots. They're going to take in CO2 through the stomates or the pores of their leaves, and they release, through the process of photosynthesis, oxygen through their leaf pores or their stomates, and sometimes you'll get water evaporating from those stomates as just a consequence of heat evaporating water. Now, plants are going to use water for its electrons. It actually needs water because it uses its electrons. And it needs CO2 because CO2 contains carbon that can be used to make sugars. So there are three reactants that are needed for photosynthesis. You need water, you need CO2, and you need sunlight. And the two products of photosynthesis are sugar and oxygen as a waste product. So the overall equation for photosynthesis is 6 CO2 plus 6 H2O to make C6H12O6 plus 6O2. So much like the cellular respiration equation, this is also a redox equation. Something's being reduced, something's being oxidized. In the case of photosynthesis, you are oxidizing water. You are stripping water of its valuable electrons, and you are reducing CO2 and splicing together those carbons to make a 6-carbon sugar, or you could just say a sugar. You're splicing CO2 together to make sugar. So photosynthesis is going to happen through two different processes. The first process is the light reaction, which requires light. So light energy is converted into chemical energy. And then the second part of photosynthesis is the light-independent reactions. Calvin cycle is going to be the light-independent cycle. And the purpose of the Calvin cycle is to make sugars. So if we look at this here, this is a chloroplast, a doubly layered organelle that we'll typically find in plants, meaning you have two phospholipid bilayers, much like the mitochondria. Within a chloroplast, you're going to have stacks of these things called thylakoids. So here's a thylakoid, this green thing. This is a cross-section of a thylakoid, so you can see on the inside, and it's stacked in what we would call grana. So your light reactions are going to happen on this pigmented thylakoid membrane. So light is going to be absorbed by the pigment on the surface of your thylakoid. This is actually what makes plants green. This is actually what makes chloroplasts green, and therefore plants green. Most plants is their photopigments that they store on their thylakoid membranes. So the purpose of your pigments is to absorb as much light energy as possible, and with that light energy absorption, we'll talk about later, what you're going to do is you're going to convert that light energy into a usable energy, like ATP, like NADPH. So As a result of the light reaction, you actually form ATP. You form NADPH. Two different types of potential energy that can be used to drive forward what we would call the light-independent reactions. So the Calvin cycle involves taking CO2 and splicing it together to make sugars. That's an endergonic pathway, an endergonic process. You're moving, you're building something from many small individual components. That's going to take energy. And so the purpose of the light reaction is to make just enough energy to fuel the Calvin cycle. So the Calvin cycle is where you're actually making the sugars, but you need to make enough energy, usable energy, in order to fuel that pathway. So the light reaction makes energy so that the Calvin cycle can take place. And just as a few things to take note of, the light reaction happens on the thylakoid membrane, or you could say the thylakoid, and the light-independent reactions happen in what we call the stroma, or that juice within the chloroplast. So let's look at the chloroplast structure. Again, this is going to be much like the mitochondria. It's going to have two phospholipid bilayers. So you have one phospholipid bilayer, and you've got another phospholipid bilayer right outside of it. Inside of that chloroplast, you have little pancake-shaped sacs called thylakoids. A stack of many thylakoids is a granum. So a singular stack is a granum. All of these stacks, as a plural, is grana. You have many grana, many stacks of thylakoids. So that stroma is going to be the liquid inside of the chloroplast that suspends that grana. The grana are going to be the stacks of thylakoid sacs. And then your thylakoid membrane is where you're going to have all of that pigment embedded. Now chlorophyll is going to be the main pigment type, and specifically chlorophyll A, which you're going to find in the reaction center of your photosystems. But you have two different types of chlorophyll that we'll talk about. You have chlorophyll A as well as chlorophyll B. Both of those are types of photopigments that are going to absorb photons moving in wavelengths in all wavelengths of light except for green, which is actually reflected. And that's why we see most plants as green, because what those photopigments are doing, what chlorophyll A and B are doing, is absorbing all wavelengths of light to use that light energy to fuel the light reaction of photosynthesis, and it's reflecting everything else, which is green. And so we see it as green. So this isn't a physics class, so we don't need to go too deep into the properties of light. But a few things could be helpful when trying to conceptualize photosynthesis. So first, we have the term photons. That just describes packets of light energy. So light will travel in the form of photons. Now, when light travels, it will travel at various wavelengths. So a wavelength can be described by the length of a wave. So here you have the peak of a wave, and here you have another peak. So the distance between this peak and this peak is a wavelength. The length between, two peaks or two troughs. Now here you have a relatively high wavelength, especially when compared to this wavelength here. From this peak to this peak, or this trough to this trough, a very short wavelength. But you have over here a lot of waves. We would call this high frequency. We would call this low frequency. So wavelengths, the length of a wave, is typically the inverse of how fast the photons move. You have high frequency, high energy on the left side, and low frequency, high wavelength on the right side. So if we look here, we've got wavelengths. spanning from very short high energy gamma rays to very long low energy radio waves. Now, all of the wavelengths of light that we can see range from about 400 to 740 nanometers. So this little sliver right here is what we could actually see. Anything outside of that, we can't see with our eyes. So it's generally considered that blue is high frequency. So blue over here is high frequency, short wavelength, and therefore high energy, whereas red is low frequency, long wavelength, and low energy. We generally associate, our biology generally associates blue light with morning time and higher wavelength, lower energy light, we associate that with nighttime. And there's a lot of research coming out recently kind of showing that viewing bluer or higher energy light towards the evening time could desynchronize our circadian clock because it signals to our brains that it is actually morning time, and we may begin to express genes associated with morning time at the wrong time of the day. So plants, as we know, most common plants are going to be typically green, and that roughly means that they're absorbing all wavelengths except for green, which is usually found between 500 and 600 nanometers in wavelength. So all wavelengths except for 500 to 600 nanometer wavelengths are absorbed by the leaf, and instead of being absorbed by the leaf, it bounces off the leaf. So all wavelengths except for green are absorbed. Green bounces off, it reflects from the leaf, and it hits our retinas and we interpret it as green. So this orange pen here is receiving white light, which contains all wavelengths of light. It's absorbing an array of different wavelengths and reflecting all of the wavelengths associated with orange. So it's reflecting that, and it's making contact with my retina. That signal is brought to my brain, and then my brain interprets this as orange. So on the topic of light and absorption, in nature, sometimes we might be able to observe what we would call fluorescence. So things like diamonds and rubies, for example, are substances that have the potential to fluoresce. What is fluorescence? So typically fluorescence is when some substance receives light energy, or you could say it absorbs light of a high frequency, short wavelength, and then emits light of a longer wavelength. So when a molecule absorbs light, it will typically move from ground state to excited state. So we have an example of an atom here with a nucleus and electrons orbiting the nucleus. So when this atom is bombarded with light, bombarded with photons, this electron will move from a lower energy state to a higher energy state. And then once it's done being bombarded with photons, this electron will jump back down to a lower energy state. And through that process, light is emitted. And we would call that fluorescence. So diamonds or rubies, for example, they can actually take in ultraviolet light that excites the electrons within the atoms, and those atoms move from a lower energy state to a higher energy state, and then when that electron moves back to its more stable lower energy state, it emits light. Okay, So now let's talk about photopigments. Like I mentioned before, chlorophyll A and chlorophyll B are the two main pigments we'll be discussing, though we have other types of pigments called accessory pigments, like carotenoids, which some are red, orange, or yellow. Things like beta carotene. Beta carotene is actually what is going to contribute to the various colors of leaves in autumn. We also have phycobilins, which are typically seen as blue. Now, the difference between chlorophyll A and chlorophyll B is actually very small. You have a methyl group attached to chlorophyll A and a carbonyl group attached to chlorophyll B. This actually gives them different properties and actually different absorption properties, and therefore different reflection properties, and therefore different visual properties. They're actually going to be slightly different in color due to this small difference. So chlorophyll A, we're typically going to associate chlorophyll A with the reaction center, which we'll talk about later, the reaction center of a photosystem. Chlorophyll B is generally considered to be an accessory pigment. Chlorophyll B is very close to chlorophyll A in that it absorbs most wavelengths except for green, though it's slightly different. Now, when you get the changes in leaf color with the changes in seasons, so your leaves have a storage of an array of different photopigments, chlorophyll A, chlorophyll B, many different types of carotenoids. Now, the sheer abundance of chlorophyll A is what makes the leaves green. That and the similar properties of all of the other photopigments. Typically, a lot of your photopigments will reflect green as well as chlorophyll A. Now, with the change in the seasons, what happens is chlorophyll A, chlorophyll B start to decompose. And what's left are things like beta carotene, which has an orangish, reddish color. Then you'll see red in the leaves, which is representative of the remaining accessory pigments. So here's a chart showing the absorbance difference between chlorophyll A, B, and carotenoids. So on the x-axis, you have the wavelength of light. So you go from a violet to a blue to a teal to like a lime green to yellow, orange, red. So if you see here, chlorophyll A absorbs a lot of purples and blues, and then it has this sharp drop right here. That's actually where chlorophyll B, which is considered to be an accessory pigment, actually picks up its absorption properties. And both chlorophyll A and B drop around this green zone here. And that's why we see most plants as green. Now we also have carotenoids, which are, all of these are working together to absorb as much light as possible, as many wavelengths as possible. So we have carotenoids that pick up over here, and carotenoids is kind of it's a family of photopigments that can range from yellow to orange to red. Chlorophyll B is going to be more of a lime green, whereas your chlorophyll A will be more of maybe a bluish green color.